Hi, I'm Dr. Jim. I thought it might be informative, since both coronavirus and influenza will be circulating at the same time, to talk about the structures of each. First, a review of the SARS-CoV-2 coronavirus. It is believed to have its origin in animals. It belongs to the genus Beta coronavirus. This virus is a single-stranded RNA virus. It has a lipid membrane. Embedded in that membrane are various proteins, including the envelope proteins, membrane proteins, and spike proteins. It has been shown that the spike protein binds to the host cell and is required for infectivity of the virus. Influenza A virus causes influenza in birds and in some animals. Influenza A belongs to the alpha influenza virus genus. Influenza A is a single-stranded, segmented RNA virus. The segmented nature of the RNA allows for the exchange of genes between different viral strains. Surrounding the RNA is a lipid bilayer. Inside the lipid, in addition to the RNA, are proteins which package and protect the RNA. Two large proteins protrude through the lipid bilayer. One protein is HA, or hemagglutinin. This protein is responsible for the binding and entry of the virus into the target cell. Na, or neuraminidase, is involved in the release of the newly synthesized virus from the target cell. These proteins are the antigens to which the host antibodies bind. Influenza A viruses are categorized into subtypes based on these two proteins. There are 16 known subtypes of HA and 9 subtypes of Na. The H1N1, H1N2, and H3N2 are the only known subtypes circulating among humans. Almost all cases of influenza A have been caused by descendants of the 1918 virus, which was an H1N1 subtype. Influenza B is only known to infect humans and seals. The structure of influenza B is similar to that of influenza A. It is also an RNA virus, which is single-stranded and segmented. The virus also contains a lipid bilayer which surrounds the RNA and proteins, just as in influenza A. Influenza B also contains hemagglutinin and neuraminidase proteins. There are two known circulating lineages of influenza B virus based on the antigenic properties of the surface hemagglutinin.